Alright, now that we know the basic process, let's figure out where this shuffling of chromosomes actually takes us. And this is, after all, one of the critical things about being a living organism, about being an organism that transmits genetic information from one generation to the next. So shuffling chromosomes is what we're going to figure out how to do. The question many of us have asked ourselves before is, why do I look different from parents and siblings, or perhaps why do I look similar to parents and siblings? And this is a question, of course, that a lot of people have asked about, and the answers weren't so easily forthcoming, because one of the things people thought for a while is that if you have mom and if you have dad and you're their offspring, then maybe you would be the average of mom and dad. Right? That could be sort of like mixing paint, right? You take white paint and you take red paint, you mix it, you get pink. But people realized very quickly that that wasn't what happened, because if that did happen and you always have an average and an average and average, then everybody would kind of look the same at the end, right? And yet here we are, many hundreds of generations after the emergence of humans, and we're still all kind of looking different. Yeah, we have similarities, but we still have very distinctive features. And so it turns out that this business of sexual reproduction, with meiosis as a key component, is helping us preserve and rearrange genetic information. In fact, there are 3 ways by which this happens. 3 ways by which you can mix things up. Now, this is remarkable because when you mix paint, well, you take the one paint, you take the other paint, you mix them up. That's one way. What else are you going to do with paint? That's really all you can do. But because of the process of meiosis, you can actually do it several ways. Some of these rearrangements happen because of the way the chromosomes move around during meiosis. And of course, then crossing over can happen. And then thirdly, there is a game of chance at the end of it. So we have random movement, we have random crossing over and exchange, and then we sort of roll the dice and figure out what happens next. And if you take all these possible combinations and you try and do a mathematical estimate for how many different ways there are to combine human chromosomes, that means the 46 chromosomes that we have available, you actually end up with over 70 trillion different combinations. Now, currently, the human population is at just a little over 7 billion, right? And when we add up all the people who ever lived since the emergence of Homo sapiens, we may just be able to get another 7 billion or so. So it's a lot of people, 14 billion, but here we're looking at 70 trillion. So the chance of having one human be exactly the same as another, based on this process, so far is basically 0. It just hasn't happened, and it's probably not going to happen anytime soon. Very close, yes, and they're twins, yes, but not identical. Never absolutely identical. Twinning is a different case, because in twins you have a different way of getting to the end, but still. So, the first line of getting genetic variation is by something called independent assortment of chromosomes. And independent assortment means that when you have one chromosome go one way, does that always carry another chromosome with it or not? And the answer is no. So chromosomes can do all kinds of things because they're floating around in this nucleus and they're not somehow attached one to the other. And these are the examples here when you look at what happens in meiosis. So let's follow the first path here. And when you look at the first path here, you see that there are chromosomes in blue and chromosomes in red. Now, traditionally, what we do here is we say the chromosomes in blue are the ones from dad, and the ones in red are the ones from mom. And so, if you have in your own cells currently some chromosomes from mom, some chromosomes from dad, then when your cells go through meiosis, when you've reached sexual maturity and they go through meiosis, they will need to line up, these tetrads will line up in the center of the cell, and they can line up such that all the chromosomes from dad are on one side, and all the chromosomes from mom are on the other side. And so if that happens and the cell divides, then you're going to end up with the chromosomes from dad in one cell line. And so these gametes at the bottom here are going to be just like all the ones from dad, and the other ones are going to be all from mom. Now, if you try to do something and you try and figure out the possibilities, let's do this with playing cards. When you have two decks of cards, let's say that some of them have the red back, right, and some of them have the blue back. And you shuffle these. And you shuffle them and you don't look at them. And then what you do is you deal them and you say, okay, one on one pile, one on the other, one on one pile, one on the other. It is possible when you do this kind of dealing that you end up, even after you shuffle them, with all the red cards in one pile and all the blue ones in another. But it's not very likely, is it? In fact, it's extremely unlikely. So even though this first one here is a possibility, it's really only a very vague possibility. It's much more likely that you have something like the second case, where some of the chromosomes from dad line up with some of the chromosomes from mom, and vice versa. In which case, you're always going to end up with some type of mixing. And that's what we mean by independent assortment of chromosomes. I think you would probably agree that if you had a dealer in Vegas or something like that, and they were dealing only blue cards to somebody, you'd probably go, well, what happened here? Right? This is not supposed to happen. Or somehow, you would suspect that there's something going on. And of course, that doesn't tend to happen in real life because they shuffle these things. They shuffle multiple decks and so on. And that's what we're doing here. We're shuffling our chromosomes. They assort independently, and that provides us with the first measure of variation. Secondly, it's this crossing over, what happens during prophase 1. We already heard that these homologous chromosomes, when they pair, they may go through a little overlap. And as a consequence, the chromosomal pieces can be exchanged. Now, this is a key event in your lives, and you didn't even know this. It's a key event of your lives because up until that point, your chromosome material is always clearly identifiable as coming from mom or from dad. But once you personally go through meiosis, that is when you end up producing chromosomes that are unique to you, because that error can only occur in you. It's not an error in the sense that it's bad, it's just an abnormal part of how things are transpiring here. So here is this tetrad, and there's chromosome from dad on the left, chromosome from mom on the right. Now we have crossing over, and there is a Greek term for you, this chiasma. Chiasma, because this is shaped like an X, that's why they called it a chiasma. That's what they observed in chromosome research. They saw that these chromosomes were actually crossing over, and they thought, how interesting. These kind of get stuck together. They call that a chiasma. And they call this a chiasma because eventually you notice that there's an actual piece of one chromosome that gets exchanged with the equivalent piece on the other. Now, when all of this is then changing and your chromosomes are being pulled apart, you end up with producing gametes that include so-called recombinant chromosomes. This one is clearly the chromosome from dad. This one is clearly the chromosome from mom. These ones are yours. So during meiosis is when your genetic material finally becomes your own. And that's the only time during your life where you are completely independent of what happened with mom's chromosomes and what happens with dad's chromosomes. This is where you come into your own genetic identity. And so it's pretty profound if you think about it. You are right now a product of your parents. But those cells that you release to create the next generation, those are then uniquely yours. And those are the only ones that are uniquely yours. Lastly, there are all kinds of random events that happen. The specific way by which crossing over occurs is pretty random. Sometimes it happens, sometimes it doesn't. It happens more frequently with long, wiggly chromosome arms than it happens with short ones. So there are all kinds of differences when it comes to this. And then on top of it, in males, we produce millions of sperm, and these millions are all different. There are thousands of different kinds of sperm. And then the question is, well, which one of these thousands is actually going to fertilize the egg, if any at all? And so, since each of these sperm cells has the same amount of energy available, has the same overall properties in terms of size and how well it glides through the liquid and all those kind of things, there is no real way to say this one is going to make it. Instead, it's just a game of chance where, well, one of them is going to make it and we don't know which one. And so as a consequence, one randomly selected sperm cell will fertilize a human egg. So this whole business becomes very randomized after a while. So you have all kinds of ways by which you can increase genetic variation, and this is really important in the grander scheme of things. Genetic variation is actually the kind of thing that keeps us variable enough as a population so that if one disease hits us, whether it's the flu, whether it's Ebola, whether it's AIDS, not everybody is going to die. There will always be some that have the right genetic variation to remain resistant. And this is why it's a good thing to have a genetically very variable population. Having said this, though, once you allow errors to sneak into a process, these errors may not always produce good things. But given how important it is to have a highly variable population, the good about this process outweighs the bad. But sometimes the bad is what comes to the forefront. So this is something to take into consideration. Most of the time, these errors, when they're not a good thing, are so small that they don't really matter. Little tiny things can go wrong, but in the grander scheme of things, you won't even see them. You still grow up to be a normal person and everything is fine. Or, perhaps, they are so fundamentally bad, so negative, that the organism is not going to be growing at all. So those would be problems that are lethal. So those are the extremes. Either whatever error is that's not working isn't going to make any difference anyway, or the error that's not working is so bad that you don't see it. What about the ones that are in between? What about if you have some kind of an error as a consequence of all these differences in how gametes are produced, and these errors are severe and they actually result in an individual with a phenotype that becomes problematic? Well, when that happens, we call that a genetic disorder. Now, there are a variety of genetic disorders, and even within genetic disorders, there is a spectrum between those that are relatively minor to those that are definitely major. For example, lactose intolerance is a genetic disorder. It's not because you don't like lactose, it's just you're missing something that would allow you to deal with lactose. And if you're missing something, that means it's a genetic issue. But you can deal with it. There is a dietary way to dealing with it. And so as a consequence, this may not be so bad. Some of the more serious things are changes in chromosome number. And chromosome number is, of course, something that's very highly conserved. Chromosome numbers in us have to be 46. And if they're not 46, then this can create a problem, because if we don't have 46, then maybe we're missing something. And that's called non-disjunction. Now normally, when you look at chromosome numbers and chromosome pairs, you are realizing that during anaphase 1, these chromosome pairs separate. So you have this tetrad, it's paired up, and then you move things away. Or during anaphase 2, you have the chromatids separate. So those are the two divisions by which these chromosomes separate. Anaphase 1 and anaphase 2. In order to accomplish this, you always need spindle fibers. And these spindle fibers are the little rope-like things that they actually latch onto the chromosomes and they pull them apart. And if you don't have a spindle fiber pulling on a chromosome, well, then it's not going anywhere, is it? If one from one side pulls and there's nothing pulling back from the other side, you're going to end up with extra chromosome material on the side that did the pulling. And so you may end up with the wrong chromosome number. As a consequence, the egg that doesn't have that extra chromosome could die. And then, of course, that becomes an issue, right? Eggs don't come in the millions. If one of the sperm dies, that's not really a big deal. But if the eggs die, then that is a problem. So the egg that lacks that chromosome may die. But what about the egg that has the extra chromosome? It's got all the genetic information it needs. In fact, it has a little extra genetic information. So it will survive. But then when that particular egg is fertilized by the sperm, then what happens? I mean, what difference does this extra chromosome actually make? And here is how you might end up with this kind of non-disjunction. There are two ways. The first one is you have non-disjunction happen in meiosis 1. So ordinarily, you would have another one of these spindle fibers come from here, but that spindle fiber isn't there. So somehow this non-disjunction means that you're transporting an entire chromosome over to the wrong cell. So here it is. Then you go through meiosis 2 and you end up with an extra, right? N plus 1. You want N. That's what you want. But you have an extra. And so here you produce, as a result of meiosis, you produce 2 gametes that have the extra and 2 that don't have enough. Well, these ones aren't going to make it. So what you produce in this case is only defective gametes with the wrong number. The second version is this. In this case, non-disjunction happens only in meiosis 2. The same thing though, this is where you're supposed to have the spindle fiber, but it's not there. And as a consequence of this, you have the same as before. Here is your N plus 1. This one won't survive, and these ones are normal. So if non-disjunction happens in meiosis 2, that's not as bad, because you do end up mostly with normal gametes, which is not a problem, and you occasionally end up with one that doesn't have the right chromosome number. So two ways by which non-disjunction can occur. In meiosis 1, it's a big problem. In meiosis 2, it's not as much of a problem. Well, the result of non-disjunction. What is the result of non-disjunction? Well, clearly, if you have the abnormal egg cell and the normal sperm cell, and this is actually how this turns out, the abnormal egg cell with the normal sperm cell is going to produce an abnormal zygote where one chromosome is too many. Now, you may, with some justification, ask, well, what does that matter? I mean, you have all the information you need, you just have a little extra. And that's a very good question, because we do see that there are some serious side effects if you have a little too much information. It's not altogether obvious for why this may happen, but we do see phenotypes where this is a serious issue. For example, Down syndrome is a genetic disorder, and the only thing that's wrong with the individual that has Down syndrome is that they have an extra chromosome 21. So here is the karyotype for this individual, and this is an individual with 2 X chromosomes. 2 X chromosomes means that she is female, and she has 1 extra chromosome 21. Now notice here, chromosome 21 is actually one of the smallest chromosomes in this entire karyotype. So yes, there is extra information, but it's only a teeny tiny bit of extra information. So why does that matter? It turns out that during development, chromosome 21 does certain things. There are genes on chromosome 21, and when these genes on 2 chromosomes number 21 act, they will lead to normal development. When you have 3 chromosome 21s produce whatever it's producing, you're actually ending up with 150% of what you need. Now, if you think about overproduction in general, you would all say, well, a little bit of overproduction isn't a problem. I mean, you think of the resources, think of manufacturing. If you make a little bit more, what's the big deal? But if you make 50% more, then that's a little bit of a problem, right? Think about it. Somebody says to you, okay, can you make me 100 copies of this? And you go, sure, and you bring them back 150 copies. Well, what are they going to say? They're going to say, well, why did you make 50 extra copies? That's a waste, right? And so it's not so much about the expense of doing this. It is that the resources from perhaps somewhere else are diverted. Maybe sometimes too much that's being produced isn't a good thing. And so as a consequence of this, you see that there are changes in the phenotype of the individual that change the features, change some of the internal workings of the body to some degree. And as I mentioned to you before, this is also where you end up with extra of the gene that produces this certain protein. It's a protein that we can call the Alzheimer protein. And because it produces the extra of the Alzheimer protein, 100% all people with Down syndrome get Alzheimer's at some point. And it's actually very interesting because there's a spectrum. When somebody has Down syndrome, there are those that are very functional. They have this genetic disorder, but they can read and write, they can have jobs, they can live by themselves, and so on. And on the other end of the extreme is where there are people who are clearly physically debilitated. They can't function by themselves. They don't have the mental development so that they can live by themselves, and they're confined to some kind of facility usually. And this is only because of this little tiny chromosome 21 and how long this error was turned on. So for those people who are clearly functioning, maybe that chromosome was only doing what it was doing for a short time, whereas in the others it was happening for a longer time. And so there is the scale of what can happen. And when you look at somebody who has pretty much all their facilities as a Down syndrome patient, and you'll find that once they get into their 50s, they will lose the ability to comprehend properly, they will lose the ability to write properly, they will lose the ability to understand properly, and that is actually a symptom of Alzheimer's, not of Down syndrome extended somehow, but it's actually Alzheimer's. So it's a very interesting way to look at that. Now, one of the questions people ask is, why do we end up with non-disjunction? I mean, it is obviously something that happens, but it doesn't happen very often. And it turns out one of the reasons for this is the age of the mother. When a mother becomes older, meiotic errors become more frequent. And that is because hormone levels tend to fluctuate more. So on this graph, you see that once you get to age 40, that's about here, the mother's age 40, then you end up with an increase to about 1%. So at age 40, the risk of having a child with Down syndrome has reached about 1%. And 1% when it comes to medical issues, that's often a threshold at which it becomes interesting or it becomes noteworthy. And so people were actually amazed that this happens. And so one of the things that they did is they tested the mothers for their hormonal output during pregnancy. And it turns out that as a woman approaches the end of her reproductive years, hormonal fluctuations increase. And this starts sooner in some women and later in others. So it's still perfectly fine if you are a mother who at age 45 or so wants to have a child. It's just that statistically the risk of having a child with Down syndrome increases because an older mother is going to have a greater likelihood of non-disjunction. So not only does that mean it's going to be more difficult for the mother to have children, because obviously some of these non-disjunction events are lethal, but it also makes it more likely to at some point have a child with some type of non-disjunction, such as Down syndrome. And it turns out that there are a variety of areas, including sex chromosomes, where you find these kinds of non-disjunction issues. So if you look at this table, for example, you see that there are some that are fairly interesting. So here you have something called Klinefelter syndrome, and that's an XXY male. So the phenotype of the individual is male. They do have this Y chromosome after all, but there is non-disjunction. And the frequency in the population is 1 in 2,000. So that's not that many, but that's not that few either. You also have XYY. This is not called a syndrome because the individual is actually normal. With XYY, perhaps there are some behavioral issues. The extra Y chromosome may increase, for example, testosterone or some things like that, and perhaps the individual is a little bit more aggressive, perhaps a little bit more liable for, likely to have something like attention deficit issues. Those kind of things are possible, but by and large, this is an individual thing, and so you basically have a normal male. Or you have this, that's called triple X syndrome. They don't call it anything here, but it's triple X is how I've heard it. That would be a normal female, and there ultimately is no real problem at all in females with that genotype. And then lastly, there's Turner syndrome, where there is no Y chromosome. So it's an X and empty, X and nothing, which means that this individual has only 45 chromosomes, whereas these all have 47. Right? That's one extra, and this one has 45. This is called Turner syndrome, and there are some clinical issues with that, and it's also something that does not happen quite as frequently in the population because it is something that has a negative impact on the individual. But sex chromosomes, that's one thing. That changes things ever so slightly, whereas as we saw, even that little tiny chromosome 21 can have a pretty profound impact. And that's the end of chapter 8.